This chapter is really the core of the unit. We know that traits can be passed down and vary, but what happens next? Last chapter we showed how inheritance causes new DNA combinations that leads to variations in traits. This chapter focuses in on natural selection, artificial selection, and genetic engineering. Students will be able to explain each of the cause and effect relationships on this graph by the end. We begin with a phenomenon that leads to a mystery. Darker colored mice are more common in some areas than other. Why is that? Up to this point, students have documented how traits vary, but now it's time to rate the trait. What makes a trait good, bad, or meh? Is a tall neck on a giraffe good? Students realize that it depends on the environment. In the next activity, students track the color of mouse fur over several generations in 2 different areas. They notice that on light-covered ground, mice are more light-colored. But when a dark lava flow covers the ground in one area, the colors shifted. Students count the mice and create a graph of their data. In the next lesson, students finish their bar graphs of mouse color. Then students look at another situation where traits varied as they follow along with Darwin's finches. Students then put the pieces together and explain what caused the traits in the pocket mouse population to shift over several generations. They discover the same thing that Darwin did, that populations adapt to their environment by passing on traits that make them more fit to survive. Survival of the fittest. Selection isn't only natural, though. At the grocery store, you can find orange and purple cauliflower, among other delicacies. And over the next few lessons, students will figure out how these came to be. In Lesson 11, students revisit the Humans and Life Unit Roadmap and discuss the new colorful cauliflower phenomenon. Then they participate in the interactive artificial selection presentation to see some examples of what humans have created and how they create it. Finally, students work in pairs to read and research artificial selection in animals or plants for a jigsaw, where each pair becomes an expert on one approach. In Lesson 12, students briefly discuss how many different artificially selected organisms did you encounter since class yesterday? Then students complete the artificial selection jigsaw from the previous lesson, including a whole class discussion. Students then compare and contrast natural selection and artificial selection in small groups using a Venn diagram so they can then contribute to the whole class synthesis Venn diagram. In Lesson 13, students do a gallery walk of yesterday's Venn diagrams and then explain the original phenomenon. People have bred orange and purple cauliflower. Then students participate in the genetic engineering presentation to address the question, what happens when we manipulate DNA directly? Students use the remainder of class to develop a simple physical model of the process of genetically engineering insulin. In Lesson 14, students finish building their models from the previous lesson and then share them with classmates in small groups. As students are working, circulate around and make sure that the models all contain 2 types of DNA, one from the human and one from the bacteria, and that the bacterial DNA is circular, and that they show a process where they remove DNA from the human and place it into the bacteria, like with these pipe cleaners. End the day with a unit concept checkpoint, one of our multiple choice assessments with questions that are specifically written to target misconceptions about the ideas in the chapter so far. With their understanding of genetic engineering so far, students might be able to explain this new phenomenon that people have engineered glow-in-the-dark mice.
Glow-in-the-dark mice?
Lesson 15 launches a new activity where students look at the impact of this science on society. We introduce the phenomenon and then have students discuss how it fits in with the rest of the unit roadmap. Then we introduce the criteria for the Risky Business GMO Research and Pitch Project, a pitch to a team of investors interested in funding a GMO. A genetically modified organism. We assign each pair of students one organism to research. They need to create a 2 to 3 minute slide presentation with 4 parts: about the GMO, a risks and benefits chart, their sales pitch about why investors should invest in the GMO, or a cautionary warning explaining why the investors should avoid it, and references. In the next lesson, students continue to research their assigned GMOs. Remind students to complete their charts and save links to websites used and to ensure that those websites are credible. Today, each team should focus on completing the risk and benefits chart with contributions from each team member. Students determine which column, risks or benefits, has more items, and they rank the factors from most important to least important. This activity really helps students think about the relative importance of each factor in order to help them formulate their position for a presentation to the fictional investors. In Lesson 17, students work on their GMO pitch presentation. Share the risky business rubric, which has criteria for each of the presentation sections, plus a participation score. In Lesson 18, students deliver their pitch presentations. The rest of the students act as the fictional board of investors. It's their decision about which GMOs to invest in. In order to make that decision, they need to take notes on each GMO, including the risks and the benefits, the team's position, and their own thoughts. After each team has presented, students turn in an exit ticket with their ultimate decision. What was the most common risk mentioned during the presentations, and which team gave the most convincing presentation? In Lesson 19, students return to the phenomenon of the glowing mice and the technology used to create them. Then they create a 3-circle Venn diagram to discuss science, engineering, and technology. The Reflections and Interconnections worksheet guides students to synthesize and reflect upon what they've learned. The exit ticket for the day asks, how might the world be different in 10 years as a result of the GMO technology you researched? This chapter we talked about slicing and dicing DNA to create new organisms. Well, next up is genetic mutations.